Hello, everyone. Welcome. We're going to give everybody a few more minutes to join us, but happy that you're here. Yeah. So are you also planning to attend the SRC?
Yes. Yeah.
Makes
Be there.
Yeah, so we had about 90 registered people, and typically we have half or so show up, so we'll see. It's a good crowd. Okay, handout questions. Well, let's slowly start. At least here in Europe, it's 8 p.m. Welcome, everyone. Happy that we're here. We had a little bit of a break from our regular ISEA webinars. Last one was just before summer, but we're back on the roll. We're very happy to have David Edwards with us today. He has two new roles this year, right? One is as a professor and head of the Math and Data Analytics Department at Citadel, the Military College of South Carolina, hence the beautiful uniform. But also, David joined ISEA this year as a board member, and he's, so I always invite all the new board members to come and give a webinar and sort of introduce themselves and show what they're working on, and a great way to sort of show themselves to our members, and great to learn a bit more about you. So that's it. Before I hand over to David, I wanted to give a quick shout out to our next webinar, which will be on the 11th of December, so just in a couple of weeks, by Christine Anderson-Cook, on asking good questions to facilitate better collaboration and enhance relationships. So please join us then as well. I think I saw that Christine's actually joining us today as well, so we look forward to that, Christine. Great to see you here. Let me, David, welcome. Thank you for joining us today. I'm going to hand over the presentation mode to you so that you can share your slides and your talk. Make presenter. There you go. And then maybe for the audience, if you have any questions, you can type them in the chat, and we'll have room for all those towards the end of the talk to discuss and ask your questions and get answers to them. So feel free to put them throughout if it's, and then I'll be moderating and managing that. David, thank you very much. The floor is all yours.
All right, thanks, Ines, and I do appreciate the invitation to give a talk, take part in this webinar series. I do think that, of course, this is a rather large project with a number of collaborators. I do think that this whole idea here of looking at experimental designs for high throughput screening, or as I got here on the slide, biological screening, really does kind of fit in nicely with the statistical engineering paradigm, because this is it's quite a large problem. It's, in some ways, it's a bit unstructured, and this methodology is kind of came about because of the problem, and maybe the need for a solution. So I'm going to talk today about these row-constrained designs for biological screening. First, just of course, mentioning the number of collaborators here, Byron Smucker with Henry Ford Health, and then everyone else on the list here is at Miami University, but you can see that there's kind of a variety of expertise. A couple of folks from the statistics department, we've got chemistry and biochemistry, bioinformatics and functional genomics, and information systems and analytics. And so there's a wide variety of expertise that kind of took place and came together for this project. There's, I guess, a number of course, applications of high throughput sequencing, high throughput screening. One of the things that I did at my former institution, we did a lot with the microbiome, the Human Microbiome Project, and in order to kind of collect that data involved a very large amount of high throughput sequencing What we're going to be looking at today is the whole idea of biological screening, and I'll kind of give some background there before delving a little bit into the statistics that's needed for this, and then the actual proposal of these row-constrained supersaturated designs. And I will do a little bit of comparison with some existing methods, and show you at the end an example of a real experiment that was conducted. It is a little bit on the smaller scale, and since this experiment, there's been some ongoing experiments that are on a much larger scale, which I think is more in the flavor of what high throughput screening is all about. So let's start with biological screening. High throughput screening, widely used in a large number of the life sciences, and here's just a few examples of that. Certainly not limited to these, but things that I know some about, and certainly plenty of things that I know absolutely nothing about as well. Functional genomics, structural biology, synthetic biology. The one that's listed last is the one that I'll mainly be talking about, is drug discovery. According to this 2018 article here, nearly 1/3 of clinical drug candidates come from high throughput screening campaigns. When I say drug candidates, I mean things that are further investigated as the potential for drugs, whether they be antibiotics or obviously something else, but a lot of drug candidates come from these high throughput screening campaigns. So I think a very important, very practical problem. Here's just an example. I'm not a biologist, and so I will probably, I can't tell you all of the mechanisms involved in some of these things here. The metallobeta lactamase inhibitors are of particular interest because they prevent bacteria from developing antibiotic resistance. So notice I'm using the word inhibitor. So things that inhibit these are of interest to prevent the bacteria from developing the resistance. And a particular enzyme of interest, you'll see this show up a little bit later, is this one called VIM2, Verona integron encoded metallobeta lactamase 2. And these sorts of enzymes, they're prevalent in bacteria that cause these nosocomial infections, those that are infections that occur during hospitalization. You hear names like MRSA, and these are quite bad infections. And in fact, these they cause infections that contributed to an estimated 50% of the COVID-19 deaths. And so we're looking at a particular enzyme here of interest. The real experiment that I'll show you towards the end of this webinar will involve this VIM2 enzyme of interest. So what are the logistics of high throughput screening? We've got these plates. You can see them on the screen here. You're seeing 3 different versions of a plate. And inside those plates, you can see, again, the 3 different versions are these wells. And the well sizes can be a little bit larger, like the well sizes there on the left, or quite small, like the well sizes on the right. So these high throughput screening microplates are these plates that have a certain number of wells. And obviously, we're going to be putting things inside of these wells. But of course, the plates themselves have varying numbers of wells. Here's just kind of a picture of an automation of injecting compounds into these plate wells. So again, I would imagine it would take a very long time for a single individual to try to pipette compounds into these individual wells. Obviously, a lot of times it's quite automated like this, where we've got a machine that is pipetting these different compounds into these different wells. And of course, once we've got these compounds in these wells, what happens next is whether, like I was saying before, it might be sequenced. Here, we're actually looking at collecting information. I think, we think of this as like bioassays, and they'll have readouts from these bioassays, and that'll be the responses that we collect. Some response from every well. So what's their proposed approach? Well, given this this situation, given the high throughput screening situation here, what we're going to do is we're going to treat that as a multi-factor experimental design problem. every biological entity, for example, these compounds, are going to be treated as factors. There can be a very large number of compounds of interest. So, you know, the example that I'll look at a little bit later specifically had 31 compounds of interest. But let's see, if I try to find, I had something in front of me just a second ago and I seem to have lost it. We're looking at over perhaps 500 compounds of interest, even more, we're 1,000 compounds of interest. And so the problem can actually get quite huge. And what we're going to do is we're going to treat these compounds as factors in an experimental design. And many of us are familiar with the usual 2-level experimental designs, the ones that we learn almost right off the bat in an experimental design course, fractional factorials, where we code things as -1s and +1s. That's how we're going to treat things here. Every biological entity, a compound, treated as a factor with 2 levels. Either the compound is absent in a well or it's present in a well. Every well will be treated as an independent experimental unit. And since the number of compounds is often much larger than the number of wells, we're going to need to use the machinery of supersaturated designs. And so I'm going to spend a few minutes talking about supersaturated designs in general, but you can kind of think of how this makes sense here. I've got these plates. You can see we're very restricted perhaps on the number of wells on the plates. But I could potentially have, you know, many compounds. And so since that number of compounds is more than the number of wells in general, we'll use supersaturated designs. And when I say supersaturated designs, I'm talking about experimental designs that have more factors than experimental runs, more columns than rows. What we're going to be talking about here is situations where we do pooling. And when I say pooling, I mean that every well is actually going to receive potentially multiple compounds. And this is a strategy that I'll talk about a little more in a second, but that's the whole idea. Every row of my experimental design is going to say whether or not a compound is present or absent in that well. And so I might be able to mix together many compounds in a single well. So let's talk a little bit about the statistical background here. What are supersaturated designs? These have been around for a while, but honestly haven't received a large amount of interest between 1962 and 1993. But then ever since the early 90s, we've seen almost a plethora or an explosion of research in supersaturated designs. Started off a little bit slowly. Most of it was focused on design construction, but since then a little bit more on design analysis. Because if you can imagine, we've got a situation where I have fewer runs than factors, so I can't even say estimate a main effects only model with these types of designs. So these designs kind of came about by Booth and Cox in 1962, stayed stagnant for a long time, and then Dennis Lin and Jeff Wu in 1993 kind of revived the interest, and it's been an increasing amount of interest since then. So here's a toy example with 6 runs and 9 factors. You can see here, I've got a design that is balanced, doesn't have to be balanced, and when I say balanced, I mean an equal number of +1s and -1s in each column. But the balance isn't required. But you see I have 9 factors, 6 runs. With only 6 runs, I certainly cannot estimate, you know, all 9 main effects. So this would be an example of a supersaturated design on a very small scale. SSDs, or supersaturated designs, generate data to estimate. We typically use the linear model. Most of the time, X is just going to be the main effects model, but I'm going to need effect sparsity to hold. If you're not familiar with that definition, effect sparsity just meaning that really only a few of the effects are actually important. Perhaps a rule of thumb is no more than 20% of the total number of effects are actually important, but we really need this principle to hold for supersaturated designs to be effective. If all of my main effects are actually important, then I certainly would never be able to uncover that with only a few runs. So again, we rely on that basic principle of effect sparsity for the success of supersaturated designs. There have been a number of proposals for how we decide which terms are important, which main effects, which factors. We kind of started with proposals related to forward selection, but kind of where we've landed over the years are some of the regularization techniques like the Danzig selector and the lasso. We're going to use the lasso in order to analyze the designs here. Since our main interest is the application, our main interest is the designs themselves, we're not really going to focus too much on the analysis strategy, and just note that we will be using the lasso to do that, a regularization technique that allows us to determine which factors out of the many are potentially important in situations where the number of factors exceeds the run size. Again, if you're a little bit unfamiliar with the lasso, we kind of minimize that usual error sums of squares, but we have a penalty term on there, that plus lambda norm of the beta vector. Okay, so you can kind of see we're actually shrinking some of our estimates down to zero. Those terms that shrink to zero the slowest tend to be the ones that we would consider to be most important. We're actually going to use a version of lasso where we center and scale the responses in the design matrix, and that's typically recommended in practice. The standard design criteria, and there are many. There are actually many design criteria for supersaturated designs. If you take a look at some of the literature that even myself and my co-authors have contributed to, we've kind of recommended some other design criteria other than this one here, the UES squared criteria. But this one here is pretty much, I don't want to say the gold standard, it's just very, very common, very popular as a way of constructing supersaturated designs. So what we're going to do is we're going to say, let L represent my design matrix with an intercept column, and then S is going to be L transpose L. That's just our information matrix, and the off diagonal elements of that information matrix we'll call Sij. Because we have fewer runs than I have columns, I can't get an orthogonal design. What the UES squared criterion does is it attempts to create a design that is as orthogonal as possible. Right? It does the best that it can to create a design that is as orthogonal as possible. So what you see here is a criterion that basically minimizes the average squared off diagonal elements of the information matrix. So again, look at the off diagonal elements of information matrix, square those to make sure they're not negative, and look at that average. We want to minimize that average. And again, if minimizing that average, the attempt again is to make the columns as orthogonal as possible. We're not going to be able to achieve that orthogonality, but very commonly used design construction criterion. Here's just another example of that information matrix for a 16 by 28 supersaturated design. 16 runs, 28 columns. So that's a little bit of background on supersaturated designs. I'll mention now going back to high throughput screening. Pooling, when I say pooling again, I mean actually putting multiple compounds in a well. Has been proposed and studied rather extensively. There's a nice review back in 2009. There's a paper from 2015 that calls pooling controversial with a long history of limited success. But then this author goes on and shows a successful example of a 2-compound per well campaign. Another example with more extensive pooling there in another paper from 2008. And related types of pooling are regularly considered as well. Again, things like in pharmacokinetics. So that's how we're going to approach this problem. We are going to allow pooling to occur. Pooling constraints comes when I say even for viable applications where pooling is fine, where I can mix compounds together in a single well, there may certainly be situations where I'm limited on the number of compounds per well. Right? Maybe I have 1,000 compounds. That doesn't mean that I can put 500 of them together in a well. So there may be, for certain reasons, limitations on how many compounds I can put together. The size of the well could be one of those reasons. the minimum concentration required for detectable compound effect. Or now we're looking at more of the biological reasons, maximum solution ionic strength beyond which compounds may precipitate. And so there could be a variety of physical constraints or reasons why I can't just mix as many compounds together as I want. So that is going to be the pooling constraint. And that is really going to be the reason why this type of experimental design that I'll be talking about is coming about. Now, a typical screening approach, and probably the one that you think makes the most sense, is one compound, one well. Right? Every well gets one compound. Take a measurement from that well, and you're looking for hits. And when I say hits, I mean things that are clearly inhibitors, or I've just forgotten what the opposite of inhibit is. But anyways, this is a very typical approach. So mostly one compound, one well. It's very simple in terms of our design and analysis. It's pretty common to just have one replicate, though the more is certainly recommended. So that's the traditional approach. There is a recent proposal that is, I'd say that's most similar to ours, is an approach that's called shifted transversal designs, or STDs for short, which seems like a slightly unfortunate name, within a procedure called pool hits. And so in this case, they construct designs that include roughly equally sized pools with as little overlap as possible. They observe each pool and determine if it's a hit or not. So that's the experimental data. It's very binary experimental data, hit or not. And they use information from this design to determine hit compounds, accounting for some possible errors. I will say that the proposed approach did not feel very statistical. And so our kind of approach is, I would say, a little more traditional in the sense of it has a statistical analysis to it. The designs, again, there are a little bit of similarities in the fact that there are constraints, but these designs are not the same as the ones that we'll be talking about. So row constrained supersaturated designs is our proposal. We're using a pilot study typically to determine a reasonable limit on the number of compounds per well. What can we actually handle, and to get an estimate of sigma. The requirement that we know sigma I will use in one of the simulation studies, but in all honesty, it is not we can relax that. In some of our simulation results where we've relaxed the knowing sigma prior to performing the experiment or analyzing the results, we get pretty much the same simulation results. So we're going to construct a supersaturated design that honors the compound per well constraint. Use the lasso to identify potential compound hits, and then you would perform a validation experiment on these potential hits. So this is our proposed approach. Here's a little bit of detail on how these designs are constructed. And in reality, you can see here that it's got the UES squared criterion in it. We're going to construct designs that minimize the standard UES squared optimality criterion. We want to maintain two levels such that the XIJs are either -1 or +1. But then the very last line there, equation 3, is the constraint, such that the sum of my rows is less than or equal to 2C minus K, where C is the maximum number of compounds per well. Okay, so it may be that I've got 100 compounds, but I can't put more than 10 together in a well, right? So C is 10 in that situation. So this additional constraint right here, simply think of it as this way. +1 is present, -1 is absent. So essentially we're constraining how many +1s can be in every row of my experimental design. We're saying row 1 can't have, say, more than 10 +1s. The rest of them can be -1s. Here's an example of some kind of the characteristics or features of a row constrained supersaturated design where I have 96 wells and 144 compounds. And what you're seeing is kind of a look at what happens as I change the constraint. So I'm always going to be looking at 96 wells, 144 compounds, excuse me. But the 4 different figures there on the x-axis, you can see the constraint. So you can see, for example, the upper left corner where we're looking at UES squared. You can see that if the constraint value is quite low, I actually have rather large values of UES squared. And as I increase that constraint, the UES squared values go down. And you can actually see that somewhere around maybe 50 or so, you can really see that that UES squared value really drop down closer to 0. The maximum off-diagonal elements, you can see once again similar thing. As I increase the row constraint, or C, you can see that that really drops rather quickly and then levels off. The constraint slack, the slack has to do with equation 3. If the slack is equal to 0, that means that it's obeyed exactly. There's equality there. Where you can kind of see what looks like a confidence interval is where we no longer have a 0 slack. What you're seeing there is actually the minimum, the average, and the maximum values of slack over a variety of values of C. And you can see that up until about the point where I hit about half of the number of compounds, I have 0 slack. And I think that makes sense, right? I mean, we're saying I'm constrained as far as how many plus ones, but then eventually we're going to reach a point where it's okay to have half plus ones and half minus ones in a row. And once we reach that point, we can, we start to not have 0 slack anymore. You can see that that increases as C increases beyond, say, K over 2. And then finally, you're looking at kind of a very small scale power analysis showing how power changes as C increases. And we're just looking at a different number of effect sizes in terms of sigma. So you can see effect sizes of 2 all the way down to 0.75. So the purple line up there at the top, those are effect sizes of 2 sigma. The reddish line, 0.75 sigma. And there's nothing surprising in terms of the ordering there. It's easier to detect a purple effect than it is to detect a red effect. I don't know why I use the colors. Easier to detect an effect size of 2 versus one of 0.75, but you'll notice that as C increases, it becomes easier and easier to detect those active effects. So the constraint really does, I guess, pose some issues when it comes to the analysis of these designs. You can see that that constraint, the more severe the constraint, the higher these off-diagonal elements are, the more difficult it might be to detect active effects. So do a little bit of comparison with some existing methods. We've got 1 compound, 1 well, which the easiest one. I'll also look at the shift transversal designs, and I'll look at the row constraint designs, analyzed with the lasso. I'm going to assess these designs via simulation using typical things like the true positive rate. That's the proportion of true hits that are ID'd as hits, or compounds that are true hit compounds ID'd as true hit compounds. And then the false positive rate, proportion of truly inactive factors that are ID'd as hits. Based on the application here, we will assume positive effect directions for any hits. We will assume a known sigma squared, which again, we can relax, and we have relaxed in some simulation studies. And I'll assume some known background noise or mean response that we can also relax. With 1 compound, 1 well, each well is treated with a single biological entity and a response obtained. Again, think of it as looking at some sort of bioassay, obtaining a readout of that. That's going to be the response. And the way that we're going to decide whether or not a compound is active or not is assuming mu sigma squared in a positive effect direction. We declare compound I active if the response that we get exceeds at background plus inverse normal times sigma. Something similar for the STD designs. After testing and assuming mu sigma squared in positive effect directions, we found that if we called wells active with very similar criterion, This did about as well as we could at balancing the true and false positive rates. But this is kind of our analysis method that we will use to determine whether or not a well is active or not. And then the proposed method 3 for the row constraint designs, I will center and scale the design matrix and the response. We'll obtain the lasso solutions for a wide range of lambda values, lambda being that tuning parameter for the lasso. We'll set any of these parameter estimates across all lambda to 0 if the estimate is smaller than, say, sigma over 8, and that choice was chosen to balance the true and false positive rates. Then we'll compute the least squares estimates for the thresholded models and choose the one that has the smallest BIC, BIC being the Bayesian information criterion. And the factors with non-zero estimates are declared hits. This strategy, for the most part, follows along those other recommended strategies for analyzing supersaturated designs with the lasso, where we look across a wide variety of tuning parameter values and select that which minimizes some information criteria. This is a more automated process, which we have to do often in simulations to compare methods. In practice, what one would typically do when they're analyzing data with the lasso is to simply look at the solution path and use the solution path to help guide which factors we're saying are active or not. We looked at a few different types of effect sizes, ranging from 0.75 sigma up to 4 sigma. So ranging from something smaller than the noise estimate up to 4 times the noise estimate. And these represent the difference in the mean response between presence and absence of a compound. We'll simulate from a linear model. Epsilon comes from normal 0, 1. We'll use 0 for inactive compounds and elements of beta for active compounds. We'll use the different effect sizes divided by 2, and then beta knots just calculated from the assumed background mean response mu. So again, we're just simulating data from a linear model, and then we'll use that data with all of the different methods and compare in terms of that true and false positive rate. The design sizes that we wanted to assess were different combinations of n equals 96 and then 96 compounds, 150 compounds, 192 compounds. And we wanted to vary the constraints of 10, 30, and 50. So again, no more than 10 compounds per well, no more than 30 compounds per well, no more than 50 compounds per well. Designs we actually had to use to accommodate the STD sizes are shown here. So I won't read these to you, but I think this is certainly one drawback or downside of the STD type designs. You couldn't just construct a design that kind of maybe best met your purposes. There was a little bit less flexibility in terms of design size construction. So you can kind of see here, we had designs of size 88, 85, 91, 92, 99, with varying sizes of numbers of factors and different constraints. Just one active factor assumed, one compound. Okay, so you can kind of see that 1 out of 96, 1 out of 150, 1 out of 192. So very strong sparsity here. One active compound assumed. What are our results look like? So in the end, you can see here the true positive rates showing up. You can see I suppose, well, I won't say the easiest ones. 96 is at the top, 150 the next, 192 next, and you can see all the way to the left is constraints of 10 up to constraints of 50. And one thing you can notice, of course, is that our proposed supersaturated designs do perform better than the STD designs or one compound, one well. One compound, one well is the green line. You can see all the way down there at the bottom left. It's the same for every single one of them. So it's not included in every plot. And that's the most difficult scenario where I have 192 compounds and 10. The constraint is 10. So that's the most difficult scenario. And again, so you can see that certainly the newly proposed designs perform better in terms of the true positive rate versus the STD or the one compound, one well design. You will obviously see the typical, as the effect size increases, true positive rates increase. You'll notice that when the effect size is the smallest, we're getting true positive rates. It can be quite low, particularly when the constraint is 10. You can see in the top left, it's about 50%. Bottom left, we're looking at around 25%. As we move further to the right, that problem kind of dissipates as I increase my C. False positive rates are actually quite low for the supersaturated design. The STD designs, you can see, are actually have, I'll say, reasonably high false positive rates. You can see that the axis only goes up to 0.16. That's not 1. There are a couple cases where the STD designs do have smaller false positive rates than the proposed supersaturated designs, and that would be for the cases where I have a constraint of 10 and 150 or 192 compounds. With that said, the false positive rates roughly around, it's less than 4% for both the SSD and the STD. Here's an example of the real experiment. It was a proof of concept experiment, a series of 4 experiments for the inhibition of this VIM2 enzyme. We looked at 4 different experiments. Again, 1 known inhibitor, 30 known inert compounds. So again, proof of concept. You can see we looked at 4 cases, 26 runs, 16 runs, and 24 runs, 31 compounds. 1 is the known inhibitor, 30 are inert. And you can see that I have some different constraints. The top 2, pretty much no constraint. C is 31. But then experiment 3, use C of 10. Experiment 4, the most severe constrained experiment, C of equal to 5. Here's the result. We are looking for, I can't find the word. It's, I won't be able to say it, but it's this LCPT-PRL that we're looking to find. And so you can see in experiment 1, that would be the largest effect size shown up on the profile plot. You can see that it shrinks to 0 the slowest. Experiment 2, pretty much the same thing. Experiment 3, we're starting to have some constraint, but we can still really easily detect this, the LCPT-PRL. I forgot how to pronounce the CPT-PRL part. But you can see in this case where I've got 24 runs, 31 compounds, and a constraint of 10, I'm still detecting that inhibitor there. What happens when things get really, really constrained? We start to see some problems. So here's a situation where I am only allowing 5 compounds per well. And while the inhibitor, the known inhibitor, does show up, it's not obvious that it's active. We can't really detect it very well. When you see a profile plot that looks like that, it's not that easy to detect what's really active there. That's one of the things we're investigating right now. But further is what exactly does this constraint to the structure of the supersaturated design? And one of the things that we've found is it does induce a large number of negative correlations, and I think it can easily cause some masking of effects. So that's one thing that we're really working on. So in general, our contributions are a statistically principled and an improved solution to what we think is an important problem in biology. Proposed a new class of row-constrained supersaturated designs that have been motivated by an actual application. The challenges, the proposed designs are more complicated. They do require a algorithmic construction and do require more material handling than 1 compound, 1 well. And then of course there's the whole idea of possibility of interaction effects. Now these haven't really been discussed before, but what if there are interactions? And that's certainly something that we're also currently working on, is the idea of using these designs in the presence of interactions. So that's a current piece of work. We're looking at a further theoretical study of the designs as a function of C. It's trying to scale them up for much larger high throughput sequencing campaigns. And as I mentioned at the onset, there are some experiments, I think, in the works actually, that involve a much larger number of compounds. How do we incorporate blocking into this? So again, there's certainly more work that can be done. And here's just kind of a snapshot of what happens when we're in the presence of interactions. So these are the new supersaturated designs in the presence of interactions. And you can kind of see here that, you know, let's find the hits when there may be interactions that are present. And you can see here when there might be problems. Once again, the more highly constrained designs cause the biggest issues. And interestingly enough, we found that synergistic interactions, those that went in the same sign as the main effects, caused the biggest problem. And strong ones, strong meaning that they, like for example, we would say a strong synergistic interaction would be one if both A and B are active, then the AB interaction is active. We would call that strong. That was actually causing the biggest problems with these designs in the case of the more severe constraints. As the constraint moves to the right, the problem starts to dissipate. So we know that there is definitely some more work to do when it comes to understanding these designs under more extreme constraints and how do we more effectively analyze them. So that's my talk. Again, I think this is an interesting topic because it's motivated from something very real. Certainly drug discovery, I think, is particularly in the world of we're dealing with antibiotic resistance and things like that, trying to discover new antibiotics is probably going to continue to be a very important topic for pharmaceutical companies. And here's a possible supersaturated design or statistical approach to better solving this problem. So anyways, that is the extent of my presentation, and I thank you for your time.
Thank you, David. Great. I had no knowledge of these kind of biological experiments, except for the fact that you see the pictures, right? I see a couple of questions coming into the chat. Happy to take more or put them in there if you have some others, but let me start with the ones that we have. I think this comes a bit from the motivation of your study. Why? So you have a number of wells in a pallet, right? Why would you not use more pallets if you want to have more experiments?
Yeah, so my understanding of this is that just the logistics of it, perhaps the time it takes, and yes, you could use multiple plates, right? Multiple plates, but each of those plates still contain just a certain number of wells. And I was saying that we might end up in situations where there's thousands of compounds of interest. So great question. It might be that, yes, absolutely. If we might be in a situation where I don't have this problem, but in situations where the ones that the ones that I've seen here where we are limited in what the logistics can handle, I don't know if it's I'm assuming it has to do with the data collection process itself. I'm assuming it has to do with the pipetter, what we can actually put in a well at a time, run through the sequencer or run through and actually collect the data. So I don't know every scenario, but I certainly, from what I've seen, it's just the limitation is what they can actually physically do in the lab.
Well, I have another question from Ricardo Rios saying one goal of DOE is to find optimum responses in the most sort of simple way and less expensive way, right? Less experiments. What do you think? Do you got that? I think is the question. Like, is this the sort of cheapest way to get the best result?
Yeah, exactly. It's imagine they're just, particularly in the case where they have many compounds. Pooling is one way to investigate as many compounds simultaneously as possible. The one well, one compound may not work when I've got thousands of them and I've got a situation where, again, I'm limited based on maybe the number of plates that I have or what the sequencer can handle. And so, yes, that's kind of the idea. I want to study as many compounds as possible. I'd like to pool as many together as I possibly can, but we may have limitations there. But I'm limited by the plate sizes. And so, yeah, I'd like to collect as much information as I can in as few experimental runs as possible.
Yeah. I hope that helped, Ricardo. If not, if I misinterpreted your question, let me know. Then another question from, what if you know from a scientific standpoint, William Ross asking, you know, you have knowledge, I don't know, you know, complex confounding interactions. Is that something that you can handle?
Yeah, so that's something that we're looking at right now, actually. What happens if these compounds interact? I mean, if we know, for example, that two compounds should not be put together in a well because that creates some sort of reaction, that's more of a, I suppose, biological reason not to put them together. But even in situations where I might have all these compounds that could potentially interact in some way, and maybe I don't really even know how they're interacting with each other, that's something that we're looking at. How would I analyze these experiments in the presence of potential large number of interactions? And currently, we don't have a great answer. If you look at the literature, there's been really nothing on interactions. My sense is they're kind of assuming no interaction. But if you talk to some of the subject matter experts, they concede that yes, there are some things that could potentially interact with these compounds. And so, yeah, I mean, that is ongoing research right now where we're trying to think about, in the presence of interactions, how to identify the active compounds. I would say that it would be great if we could also identify the active interactions, but if we're looking at 500, 1,000 compounds, then trying to even identify them can be difficult. Trying to identify the interactions even worse. But being able to identify the active compounds in the presence of the interactions, I think, is a very important problem that we're trying to solve.
Yeah. Then a question for myself, a bit out of scope of your talk, but more on a high level. We see all this news about deep learning and AI being able to do drug discovery and scientific discovery. I actually don't know if it does drug discovery, and there's Nobel Prizes for scientists using AI to do that kind of thing. How does that relate to this specific problem? Do you know? I mean, I can imagine that with a very complex model that might be able to predict which compounds you are more likely to add or something like that.
Yeah, I mean, actually, that's a good point. I would assume that regardless, at the moment at least, regardless of what AI produces, it would still need to be tested in the lab to see if we truly are getting the hits.
Reduce the number of compounds dramatically, maybe, right?
Yes, yeah, exactly. Maybe it provides us with an initial screening that can make the lab work easier or more straightforward or less burdensome. But one thing that, and I don't know if anyone else has any thoughts on this, I've been seeing more and more machine learning methods like neural networks kind of used to analyze experimental design data. So we're talking about taking very small structured experiments and perhaps just throwing a neural network at it. I've seen more and more publications that are doing this type of thing. In fact, even in some of my recent collaborations with some folks in pharmaceutical engineering, they were like, well, let's just use machine learning. I don't know what others' thoughts are on that. I currently have some hesitation of trying to analyze small structured experiments with something that really does well when we have large data. But I don't want to, kind of cast that off as a possibility, and maybe using something like a neural network would help analyze one of these high throughput supersaturated experiments better. Maybe help find some strange complex relationships that we weren't even thinking about. I don't know. I just that thought popped in my head all of a sudden about using machine learning to analyze experiments, and whether or not that's a good idea or not, I don't know.
I understand your hesitance. My natural inclination would be to think as a tool on the start, on screening your components that you want to put into your experiment to reduce that based on some knowledge in the network of this biological systems or something. But anyway, let me see. I think I have one final question if you have 2 more minutes. It's asking more, I think this is more a biology question, but can the compounds be quantitative mixtures of molecules?
You'll have to ask that one again. What was the question?
The compounds, right? Can they be mixtures of molecules? Quantitative mixtures of molecules.
I'll have to admit, I do not know the answer to that question.
Forgiven.
Forgive me, please. I do not know the answer to that. I'm not sure exactly what these compounds look like.
Oh, fair enough. Great. Well, David, thank you very much for your time and sharing your research on this. And I hope it helps them discover better, quicker drug combinations. As said before, to all in the audience, thanks for joining, and hopefully we'll see you on the 11th of December for our next webinar. Bye-bye for now. Let me see, how does this work?